Welcome back, students. This is video lesson 6 of chapter Sexual Reproduction in Flowering Plants, chapter 2 of class 12. This is the last video lesson of this chapter. After this video lesson, the chapter will be completed. And let us continue the chapter by the topic artificial hybridization. What do you mean by this technique? This is the technique used by plant breeders to get superior variety of plant by crossing two different plants having complementary good traits. What does it mean? It means that if we want to incorporate good characteristics, good traits you can say, of two different plants in a single plant to get a superior variety, then this technique must be taken. What do you mean by superior variety? A variety that has good traits, desired traits of two different plants in a single plant. And this superior variety is superior than the existing variety. It is also hybrid because it is formed by crossing two different parents, two different plants. Got it? Now, what are the steps to be taken for artificial hybridization? Got it? So, there are several steps to be taken. Let us study them one by one. The very first step is selection of parents. Here you can see parents to be selected for desired traits so that these desired traits can be incorporated in a plant, in a hybrid plant, you can say. Now, how can it be done? Selection can be done. For example, say in a kitchen garden, there are two mango plants in a close vicinity. One mango plant is going to produce good fruit set. Fruits are tasty, fruits are rich in pulp, and fruits have good flavor. Another mango plant, which lie in the close vicinity, is having a good resistancy of particular diseases, but it is not good in taste, it doesn't have good flavor, it doesn't have good pulp, but it is resistant to a particular disease. Now, here you can see there are two different contrasting, or you can say two different complementary good traits. What are they? Flavor, good taste, rich in pulp, and disease resistance. You want all these characters in a single plant, then you must go for artificial hybridization. Got it? So for that you select the two parents. Got it? Here we selected one mango plant which is good in taste, good in flavor, good in pulp, and another one which is resistant to a particular disease. Now, if the flower is bisexual, the next step is, if the flower is bisexual, then emasculate the flower. What does it mean? It means in bud condition, you just remove the anther by the help of scissors, and this process is called emasculation. So emasculation is the process of removal of anther before maturity so as to ensure cross-pollination in bisexual flowers. Got it? Why emasculation is done? Emasculation is done so that pollen grains of desired traits must be deposited on stigma. Chances of self-pollination is to be avoided. So that is why in bud condition, anther is to be removed. And after removing anther, after removing stamens, the bisexual flower becomes unisexual. What it? The bisexual flower becomes unisexual and it is now a pistillate flower. Got it? So in this way, emasculation is done. But in case of unisexual flower, emasculation is not to be done. Here you can select the pistillate flower. Okay. Now after emasculation, bagging is to be done. What do you mean by bagging? You wrap the emasculated flower or you wrap the pistillate flower by the help of butter paper or cotton cloth. Why bagging is done? Bagging is done just to avoid unwanted pollen grains to deposit on stigma. We want only our desired pollen grains to deposit on stigma. For example, we want a mango pollen grain which is resistant to disease to that mango plant which has good flavor, good taste fruits set. Okay. So here we can see this is the way how emasculation is done and bagging is done. Now, when the emasculated flower or you can say the pistillate flower gets mature. When its stigma becomes receptive, then dust the pollen grains. You can say the desired pollen grains to be dusted on the receptive stigma. But before that, you are supposed to remove the bag. Okay. Now, just after pollination, you rebag the pollinated flower. Okay. And the purpose is again the same, just to avoid unwanted pollen grains to deposit on stigma. Flowers are rebagged till the fruits begin to ripen, and the seeds of ripened fruits may contain the desired traits of two parents in a single hybrid plant. And then you can select your variety, your desired variety, which may be a superior variety than the existing varieties. Got it? Now students, come to the next topic, that is double fertilization in angiosperms. So if we talk about in a simple way, double fertilization is simply syngamy plus triple fusion. What do you mean by syngamy? Syngamy means fusion of male and female gametes. And what is triple fusion? You know that in an embryo sac there are two polar nuclei. So male gamete is going to fuse with two polar nuclei. This is what is called triple fusion. Now before that, we must know that pollen tube has two male gametes along with the tube nucleus. So pollen tube enters into one of the synergids by the help of filiform apparatus, because filiform apparatus guides the entry of pollen tube into one of the synergids. Now, two male gametes are released in the embryo sac. One male gamete fuses with an egg and resulting in formation of zygote, and this process is called syngamy. So, syngamy is fusion of male and female gametes, okay, and the product is zygote, okay. So, one male gametes out of two male gametes of pollen tube, one male gamete fuses with that of an egg and resulting in formation of zygote, and this process is called syngamy. Another male gamete fuses with the polar nuclei of central cell and resulting in formation of a triploid structure which is called primary endosperm cell, and this process is called triple fusion. Why triple fusion? Because three nuclei are going to fuse together. In certain cases, we find that the polar nuclei of the central cell, before fusing with the male gamete, they get fused themselves and a secondary nucleus is formed. To this diploid secondary nucleus, another male gamete fuses and resulting in formation of primary endosperm cell, and this process is called triple fusion. And this primary endosperm cell nucleus is triploid. What does it mean? It means that the cell of primary endosperm cell is having three sets of chromosomes. So this is what about double fertilization. Double fertilization is resulted in angiosperms. If we talk about in gymnosperms, double fertilization is not seen. There we can see endosperm, but that is haploid because it is formed before fertilization. But in case of angiosperms, endosperm is formed after fertilization. So here you can see the diagrammatic representation of double fertilization. Here you can see how pollen tube carrying two male gametes enter into one of the synergids via micropyle. Here you can see two male gametes and this is pollen tube approaching the one of the synergids. Here you can see that pollen tube enters into one of the synergids which is guided by filiform apparatus. Now here you can see that pollen tube releases two male gametes. Here two male gametes are released. One fuses with an egg resulting in formation of zygote. Another fuses with polar nuclei resulting in formation of endosperm. In this way, triple fusion resulted. Okay, and syngamy plus triple fusion is called double fertilization. Got it? So this is the way how double fertilization resulted in angiosperms. You know that endosperm development precedes embryo development. What does it mean? That first endosperm development resulted, first endosperm should be formed and then embryo development will be started. Why? Because endosperm provides nourishment to the developing embryo. So first the storage food is there and then embryo can develop by taking the food from the endosperm. So here you can see that primary endosperm cell which is formed by triple fusion, okay, undergo mitotic cell divisions and a nutritive cells are formed. Mass of nutritive cells are formed, which we call endosperm. Okay, endosperm accumulates food reserves and functions as nutritive tissue for the developing embryo because it provides nutrients to the developing embryo. So it behaves as nutritive tissue. Okay. Now, on the basis of initial stages of endosperm development, we divide endosperm into three types. Here you can see types of endosperm development. Number one is free nuclear endosperm development, and then second one is cellular endosperm development, and the third one is halobial. Now come to the first, free nuclear endosperm development. This is most common type of endosperm development. Here you can see nuclear division is not followed by cell division. In this case, in free nuclear endosperm development, nuclear division is not followed by cell division. Or in other words, we can say karyokinesis is not followed by cytokinesis. Then what results? Within a single embryo sac, you find lot many nuclei. So the cytoplasm of embryo sac becomes multinucleated. Okay. Later on, it undergoes cleavage and form multicellular, but the initial stages of nuclear divisions are not followed by cell divisions. Where do you find free nuclear endosperm development? In coconut water. So what is coconut water? Actually, it is free nuclear endosperm. Okay, the water that we take from coconut, okay, that is free nuclear endosperm, and it is highly nutritive. It is recommended by doctors during dehydration. Why? Because its osmolarity is same as that of blood plasma osmolarity. Got it? And what is the white kernel of the coconut that we cut and eat? That is multicellular endosperm. It becomes later on multicellular. Before that, it was free nuclear endosperm. Later on, cell division resulted in and formation of kernel of the coconut, which is multicellular. Now, students, come to the next. Cellular endosperm development. Here you can find every nuclear division is followed by cell division. In other words, you can say karyokinesis is followed by cytokinesis. This can be seen in Dhatura, in Petunia, in Balsam. Here you can see cellular endosperm development. Now another is halobial. Okay, so halobial is that first nuclear division is followed by cell division. So primary endosperm nucleus divides, okay, and then cell also divides into two. Now further nuclear divisions are not followed by cell divisions. Okay, so you can find that the subsequent nuclear divisions are not followed by cell division. So within two cells you find lot many free nuclei. Okay, and where do you find them? You can find them in order halobials and in most of the monocots. Now students, come to next, that if we see seeds, in some seeds you find that endosperm may not be seen, may not be present, and in some seeds you find endosperm is there. Okay, so how come it be possible? So here you can see that those seeds in which endosperm is totally consumed by an embryo during its development, no endosperm is left within the seed. So such seeds are called non-endospermic seeds. They don't have endosperm because the endosperm is totally consumed by the developing embryo. So examples are pea, groundnuts. These are certain examples. If we talk about endospermic seeds, so here endosperm is not totally consumed by the developing embryo. It is left within the seed. So the seed must have endosperm. Okay. Even after embryo gets mature, you can find endosperm. Okay. So what are the examples? Maize, coconut, rice, wheat, etc. So these are certain examples of endospermic seeds. Got it? Now come to the next. Here you can find here development in dicot. Okay, so how dicot embryo development resulted. Okay, now here before studying the development of embryo in dicots, you must know the structure of a fertilized embryo sac, because in this embryo sac development of embryo resulted. Okay, so this is what is different from mature embryo sac. Okay, mature embryo sac is 7-celled 8 nucleate, where at the antipodal ends, at the chalagial end you find 3 antipodals, at the micropylar end you find three egg apparatus cells, two synergids and one egg, and at the center you find central cell, and within the central cell you find two polar nuclei. But because this is a fertilized embryo sac, so labeling should be changed. Okay, so what are the labelings? Degenerating synergids. Egg is going to convert into zygote. So in place of egg, you are supposed to label zygote. In place of central cell, you are supposed to label primary endosperm cell. In place of polar nuclei, you are supposed to label primary endosperm nucleus. In place of antipodals, you are supposed to write degenerating antipodals, because synergids and antipodals, they degenerate after fertilization. And this is zygote, which is going to convert into embryo. And this is endosperm, okay, which provides nourishment to the developing embryo. Got it? It is primary endosperm nucleus. It divides either by free nuclear or by cellular or by halobial, and then it's going to nourish the developing embryo. So embryo development in dicot consists of these stages. What are they? Proembryo, globular embryo, heart-shaped embryo, and mature embryo. So let us discuss all of these stages one by one. So here you can see, students, this is the diagram taken from Truman's publication. Okay, Elementary Biology, Truman's publication. So this diagram is taken from that book, and certain labelings are mentioned over here. Okay, now here you can see this is zygote. Okay, and this is the micropylar end of the zygote, and this is the chalazel end of the zygote. Now before cell division, before division of zygote, it elongates. Okay, and when it elongates, it divides by transverse wall. Here you can see the transverse wall. So due to this transverse wall cell division, this zygote divides into two. This larger one towards the micropylar end is called suspensor cell. Okay, it is also called basal cell. Okay, and this chalazel end, the small cell, this is called what? Embryonal cell or terminal cell. Now come to this suspensor cell. So this suspensor cell divides transversely several times, about 6 to 8 times, and resulting in formation of long suspensor. Here you can see it is going to convert into a long suspensor. Here you can see, okay, a long suspensor is seen. Okay, 6 to 8 tiers of cells are arranged because they are divided by transverse wall, transverse cell division. Okay, and resulting into formation of a long suspensor. Now the last cell of the suspensor, okay, or you can say the first cell of the suspensor towards the micropylar end. Okay, it enlarges, it becomes vacuolated, and it behaves as haustorium or haustorial cell. Okay, now first of all, let us discuss what is the role of suspensor. This suspensor pushes the embryo towards the endosperm so that embryo can derive nutrients from the endosperm. Got it? While the role of haustoria is that it helps in attachment of embryo to the embryo sac. Now come to the basal cell or the embryonal cell. This one, this terminal cell or the embryonal cell. This is the terminal cell, which is also called embryonal cell, which lies towards the chalazel end. Okay. Now this cell, this embryonal cell undergoes two vertical divisions and one transverse division. Two vertical divisions and one transverse division, resulting in formation of octant stage. Here this one is the octant stage. Two vertical division and one transverse division, resulting in formation of 8 cell stage embryo, which is called octant stage. And here the cells are arranged in two tiers. This is the upper tier and this one is the lower tier. Here you can see, here the cursor is here. Here this one is the, here you can see that embryo or at octant stage is arranged in two tiers. Okay, the upper and the lower. Clear? The terminal tier towards the chalazel end. Here you can see the terminal tier of cells towards the chalazel end. This one is the terminal tier. Okay, towards the chalazel end. Finally form cotyledon and plumule. Here you can see this is going to convert into cotyledons and plumules. Okay, cotyledons and plumule. Got it? While this one, the basal tier, the hypobasal towards the micropylar end, this one is going to produce hypocotyle except its tip. Why? Because tip is derived from the hypophysis. What is hypophysis? You can see that this is the suspensor cell. the cell which is attached with the embryonal cell here, this one, the embryo, this one, okay, is called hypophysis. And the fate of hypophysis is that it is going to convert into root tip and root cap. So it gives root tip and root cap. Got it? And here you can see this is the octant stage, divide periclinally. Now it undergoes periclinal division, parallel to the organ, parallel to the cell wall, you can say. Okay, got it? So here it divides periclinally, okay, and resulting in formation of what? Here you can see the outermost layer is called dermatogen. Okay, so the outermost layer is called protoderm or dermatogen. Okay, what is the fate of dermatogen? It gives epidermis of the embryo. And then you can find ground meristem, this one. Okay, this one is called ground meristem. It is going to give cortex and pith of an embryo. Okay, and here you can find procambium. Okay, this is going to give vascular strand. Okay, so in this way tissue development resulted. Okay, here you can see the tissue differentiation resulted, division of labor started over here. Okay, now this is what is called globular embryo. What is it called? Globular embryo. Got it? It looks like globe, so it is globular embryo. Now this globular embryo is going to convert into heart-shaped embryo. Come to this point, half. This one is F1, heart-shaped embryo. This is heart-shaped. Okay, why this heart-shaped embryo is seen? Because dicot embryo has two cotyledons. That is why a heart-shaped structure is seen, so that two cotyledons can be formed. Now this you can see a mature embryo. Here you can see the G1. This is the mature embryo. This one is the suspensor which pushes the embryo towards the endosperm. This is the radicle. Okay. And here you can see the plumule. Okay. And two cotyledons. Now this is the structure of a typical dicot embryo. Okay. The H1 you can see. This is what is a typical dicot embryo. Here you can see that embryonal axis. Okay. It has an embryonal axis. Okay. And the mid of the embryonal axis is called cotyledonary node. Okay, and from cotyledonary node, okay, the portion of cotyledonary node, okay, is called hypocotyle. Above that level is plumule, and the portion of the embryo below the cotyledonary node is called hypocotyle. Okay, so we will discuss the diagram in detail later on. Okay, now here you can see the development of embryo in monocots. Here you can see the difference between dicot embryo and monocot embryo. All the stages are same, except here you can't see the heart-shaped embryo because monocot has only one cotyledon, so heart-shaped embryo is not seen. Here you can see proembryo, here you can see globular embryo, here you can see mature embryo, but here heart-shaped embryo is not seen. Number one difference. Number two difference between dicot embryo development and monocot development is that here you can't see long suspensor. Here only 3 to 4 suspensor cells are present. There you find 6 to 8 long suspensor. Here you can find not more than 3 or 4 suspensor cells. Okay, so these are the main differences between dicot development and monocot embryo development. Again, I am repeating, in monocots you can't see heart-shaped embryo, number one, and number two, suspensor is not more than 3 to 4 cell stage long. Got it? While in dicots you can see heart-shaped embryo because two cotyledons to be produced, and suspensor is 8 to up to 8 tiers of cells are there in the suspensor. Got it? It is long. Now here you can see this is fertilized ovule, which is also called seed. Okay, and here you can see the micropyle. Here you can see the hostorium, which helps the embryo to attach with the embryo sac. Here it is attached with the embryo sac. Here you can see the cotyledons. Got it? And how the suspensor pushes the embryo towards the endosperm. Here you can see this is the endosperm. These are the two cotyledons. This is the suspensor, and this is the hostorium. So this is how the embryo grows in the ovule. Okay, so this is what the fertilized ovule showing embryo and the endosperm. This is the downloaded diagram. Now here you can see the structure of dicot embryo. So here you can see that the embryonal axis, this is the embryonal axis. I'm passing the embryonal axis which passes through the center, to the through the axis of the embryo. Here you can see. So the mid of the embryonal axis is called cotyledonary node, from where the cotyledons arises. Got it? Now say this is a cotyledonary node. The portion of the embryo above the cotyledonary node is called epicotyle, which ends with plumule. Got it? And the portion of the embryo below the cotyledonary node, here, say this is the cotyledonary node. So the portion of embryo below the cotyledonary node is called hypocotyle, and it ends with radicle and root cap. Got it? So in this way, your dicot embryo is seen. Okay, this is the diagram of dicot embryo. Got it? It is divided into epicotyle and hypocotyle. Epicotyle ends with plumule, hypocotyle ends with radicle, which is covered by a root cap, and it has two cotyledons. So this is dicot embryo. Now come to the monocot embryo. In monocot, you know that there is only one cotyledon seen. Okay, and in grass family, you find that scutellum is somewhat shield shaped. Okay, so here shield shaped cotyledon is seen, situated lateral to the embryonal axis. This is the embryonal axis, and it is situated lateral to the embryonal axis. And here you can find a rudimentary second cotyledon. Here you can see the rudimentary second cotyledon, which is called epiblast. This one. So what is epiblast? Rudimentary second cotyledon. So second cotyledon here is not functional. It is functionless. It is rudimentary. It arises, but it doesn't develop. Okay. So this is what is called epiblast. Okay. Now here you can see that at its lower end, the embryonal axis has the radicle. Here you can see. Okay. Here you can see radicle and root cap. If we talk about the fate of radicle, it gives root system, okay, of the plant body, and this radicle is covered by an undifferentiated sheet which we call coleorhiza. Okay, here you can see the coleorhiza. Okay, this one is the coleorhiza. Okay, which protects radicle and root cap. Okay, this is the sheet, undifferentiated sheet, which covers radicle and root cap. And the portion of the undifferentiated sheet which covers the shoot apex, the leaf primordia, okay, is called coleoptile. Got it? So here you can see the coleoptile, which covers the shoot apex consisting of leaf primordia. Got it? So in this way, here we can see that only one cotyledon is present, which is called scutellum. Okay, and scutellum here is shield shaped, situated lateral to the embryonal axis. There is one more monocot in monocots. There is one more, you can say cotyledon is seen, but that is not functional. That is rudimentary, and that we call epiblast. Okay, so here you can see that leaf primordia and shoot apex is covered by undifferentiated sheet, protected by undifferentiated sheet, which is called coleoptile, while the radicle and root cap is covered by another undifferentiated sheet, which is called coleoptile. This is coleorhiza. Sorry. Okay, so shoot apex is covered by coleoptile, and root is covered by coleorhiza. Okay, the precursor of root system, radicle and root cap, is covered by coleorhiza. Okay, so in this way we can find that there are different structures present in the monocot embryo. Now students come to pre-fertilization structures, which are going to convert into different structures after fertilization. So here in the first column you can see pre-fertilization structures, and in the second column you find post-fertilization structures. So here you can see that ovary is the pre-fertilization structure, which is going to convert into fruit after fertilization. Ovary wall is going to convert into pericarp, which is also called fruit wall, and it may be divided into epicarp, mesocarp, and endocarp, which is very much clearly visible in case of mango. Okay, you eat mesocarp of mango. Okay, and the hard seed coat is covered by, you can see the seed coats. After seed coats, you find a hard layer that is called endocarp, and the covering of the outermost covering that you generally throw is called epicarp. Okay, in pickles you use it, but when we take raw mangoes or the ripe mangoes, then you discard epicarp. You eat only mesocarp. Got it? Now ovule is going to convert into seed. Funicle is going to convert seed stalk. Hilum remain as hilum. It looks like a scar when stalk gets detached, then a scar-like structure is seen in the seed. That is called hilum. Micropyle remain as micropyle, through which water and dissolved oxygen enters into the seed so that embryo can develop. Now integuments is going to convert into seed coat. Outer integument is going to convert into testa. Inner integument is going to convert into tegument. New cellars generally degenerate, but in certain cases, new cellars persist. Okay, and that we call perisperm. Embryo sac cells. Okay, antibodies degenerate. Egg cell is going to convert into zygote and then embryo. Secondary nucleus is going to convert into endosperm, and antibodies degenerate. And what about sepals petals? They wither off, they shed off. But in certain cases, you can find sepals persist. For example, Solanaceae family, for example, petunia, for example, tomatoes, chilies, okay, capsicum. There you find that sepals persist even after formation of fruit. Clear? Now come to next. Here you can see this is the diagram given in the NCRT book. Okay, so here you can see here endospermic seeds and non-endospermic seeds are shown. So this one is the non-endospermic seed and rest are the endospermic seed. So these two are the seeds of monocots. Okay, these are the seeds of dicots. The upper one are dicot seed and the lower ones are monocot seeds. Got it? Now here you come to the non-endospermic seed. This is the dicot seed, pea seed. Here you can see two cotyledons are there, and within that you find an embryo. And we already have studied the structure of dicot embryo right now. Okay, that how it is divided into hypocotyle and epicotyle, and then how the two cotyledons are present that we have seen right now. Okay, now here you can see this is the seed of castor. This is an endospermic seed, and this is also an example of perispermic seed having perisperm. Okay. Now here you can see this is a endosperm, and just outer layer of endosperm nucellus persist that we call perisperm. Got it? And here you can see these are the cotyledons. Okay. These are the cotyledons. Okay. So this is what is castor seed, which is endospermic seed, and it is dicot seed. This one is also dicot. Okay. Now come to the onion seed. Okay. This is monocot seed. Okay. Here you can see this is the endosperm. Here you can see the embryo. Embryo is curved and embedded in the endosperm. Here you find the seed coats. Okay. And this is what is onion seed, which is endospermic seed. Castor is endospermic, onion is endospermic, but pea is non-endospermic. Now come to this. This is very important diagram if you talk about the board exam. So here this is what is called maize seed. This is endospermic seed. This is monocot seed, and this is the maize grain, you can say. Okay, this is caryopsis type of seed. What do you mean by caryopsis? Caryopsis means where the fruit wall and the seed pores both get fused together. Okay, so pericarp, that is fruit wall, and the seed pods are fused together. Okay, there is no gap between the two. They are fused together. So such seeds are called caryopsis. So here you can find this is the maize grain. Okay, here you can see. Okay, it can be said as fruit, it can be said as seed. So we call it grain. Okay, so here you can see that this one. Here you can see this is endosperm. Okay, and it is covered by a proteinaceous layer that we call aleurone layer. Okay, got it? And here is the endosperm. This one is the endosperm. Okay, and here you can see scutellum. This is cotyledon, shield shaped cotyledon, which is shifted lateral to the embryonal axis. And here you find the embryo, and embryo structure we already discussed in monocots, that plumule is covered by undifferentiated sheet, which is called coleoptile, and radicle is covered by undifferentiated sheet, which is called coleorhiza. Now here you can see in between aleurone layer and between scutellum there is a layer called epithelial layer. This layer secretes gibberellic acid so that nutrients can become active, they get mobile, and embryo can get the nutrients, and embryo can undergo divisions. Okay, so in this way you can see the structure of the endospermic seed in case of maize, in case of monocot. Okay, so here you can see this is a caryopsis type of seed. I repeat, in caryopsis, seed coat and fruit wall both get fused together. Okay, there is no gap between the two. Now come to the next. Here you can see these are all what I have explained already. Okay, so you can see that seeds are actually fertilized ovules. Okay, and they consist of seed coat, cotyledon, and embryo axis. Seed coats are divided into testa, which is formed from outer integument. It is thick, it is tough, and it is brownish. And inner integument is going to convert into tegment, which is inner, which is thin, which is papery and transparent. Mature seed may be albuminous, which we call endospermic seed, or non-albuminous, which is also called non-endospermic seed. Albuminous means that endosperm is left within the seed. It is not totally consumed by the developing embryo. And here you can see the examples: wheat, maize, barley, castor, coconut, sunflower. While in case of non-albuminous seed, the other name is non-endospermic seed or X-albuminous. There you can't find any residual endosperm because it is completely consumed by the developing embryo. And you can see pea, gram, bean, groundnut. These are non-endospermic seed or non-albuminous seeds. In certain cases, you find that nucellus persist. Okay, so you can see remnants of nucellus. Okay, persistent nucellus, remnant of nucellus persist in certain cases, in certain seeds. And that remnant, that residual persistent nucellus is called perisperm. And where do you find them? We find them in black pepper, in beet, in cocoa, in coffee, in castor, etc. So these are examples where you find perisperm. What is it? Residual persistent nucellus. And what is nucellus? Nucellus is simple parenchymatous tissue of an ovule, of an young ovule. Okay, you find that ovule body is made up of simple parenchymatous tissue that we call nucellus. Generally, this nucellus degenerate, but here you can see it persists. Its remnants can be seen. Okay, and that is called perisperm. Examples are already mentioned. Now come to the next, seed dormancy. Here you can see as embryo reaches the maturity, its further growth is suspended due to development of growth inhibitors. Okay, and abscission of funicle or changes in integuments resulted. The cells of integument lose their protoplasm. They develop into thick and impermeable walls. The integuments thus get converted into seed coats that we already have discussed, testa and tegment. Now the moisture content of the seed decreases and reaches up to 10 to 15. Okay, and in this dry condition, seed can be stored, okay, and can be consumed throughout the year. And that inactive stage of embryo is called dormancy. What is dormancy? Seeds are unable to germinate even after providing suitable conditions. There are several reasons for this. Embryo may be immature because of accumulation of some growth hormones. Okay, that is why seed is not going to germinate. Okay, the micropyle remain as micropyle, okay, of the seed where dissolved oxygen, water, and minerals enter. Okay, so that seed can be germinated. Seed dormancy help plants to cope with the adverse environmental conditions. So seed dormancy is must for the plant so that they can cope during their adverse environmental conditions. When they don't have seasons to grow, they can be stored their life in the form of seeds. And there is seed dormancy is also important for us. How come? Because when the seeds enter into dormant stage, we can store them and we can consume them throughout the year, and afterwards we can raise them, okay, as crops in next season, next year. Okay, so these are the basis of agriculture also. Now here you can see true fruit, false fruit, and parthenocarpic fruit. What are true fruits? Those fruits which are formed from fertilized ovary are called true fruits. Example, mango. Okay, mango, grapes, these are true fruits. Now, false fruit. Along with ovary, other floral parts also take part in the formation of fruit. Such fruits are called false fruit. For example, in apple, thalamus take part in the formation of food. Here you can see the diagram. This is an apple, longitudinal section of apple, and this is the cross section of apple. Here you can see that along with ovary, this is the ovary. Here you can see this is the ovary where seed is present. So this is not the edible portion. Though ovary take part in the formation of fruit, but it is not utilized. Okay. So here thalamus take part in the formation of fruit, which is the edible portion over here. This is the thalamus which we eat, and this portion we throw. Okay. So here along with ovary, thalamus also take part in the formation of fruit. Such fruits are called false fruit. And parthenocarpic fruits are seedless fruits, those fruits which are formed without fertilization. So grapes are natural parthenocarpic, banana are natural parthenocarpic. You can make any other fruit as parthenocarpic except few where seeds are edible. For example, pomegranate. You can't make pomegranate what? Seedless. Okay, or you can say parthenocarpic because we eat seeds of pomegranate. Okay, so in this way, many fruits can be made parthenocarpic if you want, if you don't want to eat their seeds. For example, papaya, for example, watermelon, etc. By chemical treatment, by hormonal treatments. Got it? Now here you can see significance of seeds and fruits. So to protect the seed and thus the delicate embryo of the seed, fruits are very helpful. Seeds can be easily dispersed to distant places because seeds and fruits are the food of animals and birds. So they take them, they consume them, and throw their seeds uh, at different places, distant places. Seeds and fruits also provide nutrition. Okay, and to germinating seeds and developing seedling, fruit provide nourishment, nutrients. Seeds and fruits are also source of food. It is source of proteins, oil, organic acids, vitamins, minerals, and sugar for humans. Seeds of fruit are the basis of agriculture. Seeds and fruits are products of sexual reproduction, so variations can be seen, right? Now here you can see some facts about seeds. Seeds may be viable. Viable means they are capable to germinate. Okay, they are able to germinate. Okay, so for some 2000 years. Okay, this is what is an exception. Okay, here you can see seeds of Phoenix dactylifera. It is khajur, you know, Phoenix dactylifera. Phoenix dactylifera. It have been recovered during archaeological excavation of King Herod's palace near Dead Sea, and this seed become viable up to 2000 years. Okay, now you can see another example, Lupinus arcticus. Okay, it is taken from Arctic tundra. This seed is germinated after 10,000 years. Okay, and it gave plant, it gave flower, and it bore fruits. Got it? So in this way, you can see that even after 10,000 years, the seeds are viable. They are alive, you can say. Now, orchid fruits contain thousands of tiny seeds. Other examples where you can see thousands of seeds, Orobanche, striga, these are parasitic species, plant species. There you find that fruits have millions of seeds. Got it? And you can see fig tree seed. It is very, very tiny. And from that tiny seed you can get huge fig tree. So these are some facts about seeds. Now students come to apomixis. A few flowering plants, for example Asteraceae family grasses, you can say, have evolved a mechanism to produce seeds without fertilization. Here seeds are produced without fertilization. This is what is called apomixis. Okay, so you can say in other words, apomixis is a form of asexual reproduction that mimics sexual reproduction. Got it? Why? Because seeds are the product of sexual reproduction. So seeds are produced, but without fertilization, without you can say involvement of two different parents. Here, asexual reproduction is involved to give rise to seeds. So there are several methods of apoplectic development in seeds. Two methods are recurrent agamospermy and adventive embryony. So if we talk about agamospermy, it is the formation of seed that has an embryo formed without meiosis and syngam, without fusion of male and female gametes and without meiosis. Okay, you can get seeds. So agamospermy is divided into non-recurrent agamospermy and recurrent agamospermy. Now first of all discuss non-recurrent agamospermy. The embryo is haploid here and the seeds having it is non-viable. Seeds are not going to germinate. Okay, but in case of recurrent agamospermy, all the cells of embryo sac are diploid, as it is formed directly either from nucellus, which is diploid cell, and this is called apospory, or diploid megaspore mother cell. Here, the diploid megaspore mother cell is not going into meiosis. Okay, so it remains diploid and it enters into embryonic stage. So this is what is called diplospory. The diploid egg as well as other diploid cells of embryo sac can grow into normal embryos. So this is the viable seed. Okay, here viable seeds are produced in recurrent agamospermy. Now students, come to the significance of apomixis. Hybrid seeds have to be produced every year. Why? Because seeds collected from hybrid plant, if we sow them, they do not maintain good characters of hybrid plant because their genes get segregated. Got it? So production of hybrid seed every year is very costly. Okay, and it increases the crop production. This can be avoided if apomixis can be introduced in the hybrid seeds. As apomixis is genetically controlled, therefore scientists are working on studying the apomictic seeds so that they can they can make they can introduce the apomictic genes in those plants which are produced for good characters, or you can say they can introduce those genes in hybrid plants so that they get seeds, apomictic seeds, in which no segregation resulted and the parental good characters can be retained. Got it? Now, embryos formed through apomixis are genetically free from infections. So, these are some of the significances of apomixis. Now, students, come to the last point of this chapter. Formation of embryo directly from the diploid egg without fertilization is called parthenogenesis. So, this is called diploid parthenogenesis. Okay, example, apple, popo is a type of grass. Okay, so here in this topic we are going to study polyembryony. As the name indicates, poly means many embryo. So within seed you generally find one embryo, but here in certain seeds we find more than one embryos within a seed, within a single seed. Okay, so this is because of adventive embryony. An embryo develops directly from a diploid cell other than egg cell. Okay, maybe from nucellus cell, maybe from integuments. There you can see in citrus, in apantia. In certain cases you find more than one nucellus cell becomes special, okay, and behave as megaspore mother cell, and they get converted into embryo after fertilization. Got it? So more than one embryo sacs are formed in the seed, so resulting in formation of more than one embryo. So there are different ways of polyembryony. Got it? Now, in gymnosperms, polyembryony can also occur due to cleavage of growing embryo. This is called cleavage polyembryony. So, occurrence of polyembryony due to fertilization of more than one egg is called simple polyembryony, while formation of extra embryos through sporophytic budding is called adventive polyembryony. So, polyembryony is common in onion, groundnut, mango, lemon, orange, etc. So this is what about this chapter. So here we have completed the chapter. Okay, and thank you very much.